Okay, back with photosynthesis. Photosynthesis part 2. It should be lecture video 5. Let's get into the Calvin cycle, and we will then be done. So, first step had us looking at the light reactions, where we were essentially just gathering some energy in the form of ATP and high energy electrons in NADPH. And now we're actually going to put those things to work. So what are we doing in the Calvin cycle? This is where the sugar actually gets built. Remember, that is the ultimate goal of photosynthesis, is to make sugar. And so this is where that's actually going to happen. This is where CO2 is going to get used. And this is where the high energy electrons and ATP that we made are going to come into play as well. It's going to be split into 3 main phases, which you can see in this cycle. It's carbon fixation, reduction, and then regeneration of RUBP. So I'm going to start talking about carbon fixation. So carbon fixation means attaching carbons together, fixing them together. This is the major step and goal. This is the magic that turns gaseous carbon in the form of carbon dioxide into solid wood and glucose. This is what trees do. It's the magic of taking carbon dioxide from our atmosphere and turning it into plant matter. So essentially what's going to happen is that we're going to take 3 carbon dioxide molecules, which have to be breathed in from the outside by the plant, and we are going to join them. We combine them with 3 of these RUBP molecules. There's an enzyme called Rubisco that does this. If there's one enzyme to know from photosynthesis, it's probably Rubisco. It comes up a good bit as an example, so just being really familiar with it can be helpful on the exam. But essentially all we're doing is Rubisco is just going to attach a carbon to one of those 5 carbon RUBPs, and it's going to make briefly this 6 carbon molecule that you can see. Let me put on my laser pointer. Wow, that you can see here with the 2 phosphates on the end. The 2 phosphates are going to make it pretty unstable, and so it's going to quickly break up into 2 3-carbon molecules with a phosphate group on the end. Again, the phosphate kind of destabilizes it and just makes it more willing to react. I can get rid of my laser pointer. So all we've done here is essentially take a carbon off of CO2 and attach it to a bigger carbon molecule. So we've got some 3 carbon molecules going. The next step is a reduction phase. So this is where we're going to actually use some of that energy that we made in the light reactions, where NADPH is going to come in, and it's going to essentially give up its electrons to those carbon compounds we had just made, the 3-PGA. It's also going to use ATP to do this because it needs a bit more energy. And it's going to convert those 6 3-PGA molecules, and it's going to turn them into a G3P molecule. Well, it's going to turn them into 6 G3P molecules. So 1 ATP and 1 NADPH per. Of those 6 molecules, though, only one of them is going to go and actually exit the cycle and go on to make glucose. We've made G3P, and that's going to go off, one of them is going to go off to make glucose, but 5 of them need to be recycled and reused in this cycle to keep it going, so we can keep turning this cycle over and over again. And you'll see why in just a second. Because now we are at the regeneration of RUBP, that molecule that we needed to react with carbon dioxide right at the beginning. And to make 3 of those 5 carbon molecules, I need 15 total carbons. Thankfully, we've kept around 5 of the G3P, which have 3 carbons each, so I get my 15 carbon from there. These are just going to use a little bit more ATP, and they just undergo a conversion. You don't need to worry about it to become that starting molecule again. That'll continue to react. It'll again go through, react with CO2, and the cycle will continue. So, a little faster. Ultimately, what happens to that G3P? Remember in this step, we had a G3P, and it left to go off to make glucose. It left to go do other things. And we had to keep 5 of them around so we could regenerate and keep the cycle going. So what happens to that one G3P that is released? Well, we only get that one G3P every 3 turns of the cycle. When we were going through this, we were looking at it with 3 carbon dioxides going in. In reality, it would be 1 at a time, and each one is a single turn, but we needed 3. So it takes 3 whole cycles of this Calvin cycle to make 1 G3P. But after it's made that G3P, it's going to be used for a ton of stuff. We tend to just think of photosynthesis as making sugar and making glucose, and it does, and that's great. But it's also involved in making fatty acids, so helping to make your cell membranes. It's also involved in amino acid synthesis, so we can use it to help make amino acids. We can obviously make glucose, but we can turn that into other sugars as well. It's not exclusively glucose. It can be other molecules as well. But to make a single molecule of glucose, you need the Calvin cycle to go around 6 times, or you need 6 total turns of the Calvin cycle to get the single G3P that you can extract. So what happens to that molecule is a little less clear when we talk about respiration. It's kind of just, it gets broken down into ATP and everything's great. In this case, the ultimate end point of photosynthesis really depends. It's not always clear. Most of the time we're going to think of it in terms of creating sugar and creating energy, but just know that it can also be used for these other purposes as well. And with that, we have finished Unit 3 lecture. The lecture material is a little shorter for this, but it's dense. There's a lot going on in it and a lot you have to remember. So we'll practice. Let me know if you have questions, and I'll see you later, skaters.